Welcome to this episode of Eureka, and we are going to have a very interesting discussion with Dr. Apurva Sarin, who is the Dean of Institute of Stem Cell Research and Regenerative Medicine. Stem cells. Many people think that's going to be the future of medicine, particularly in terms of organ transplant. Today, if your organ is failing, you look for some donor. Tomorrow, they say, if the stem cell research yields us the result, you can regenerate your own organ. But then there are others who pointed out that perhaps there is unknown danger lying behind this attempt. We will understand about stem cell research and this institute and how India is progressing in this area in this conversation. But before that, let's take a quick look at her brief profile.
Presently, Professor Apurva Sarin is heading the InStem Institute in Bengaluru, where scientists and clinicians work in team-based approaches to address outstanding questions in stem cell biology with implications for human health. Professor Sarin obtained her master's in zoology from Delhi University and PhD from JNU. After that, she started working as an assistant professor at Indian Institute of Science in Bengaluru. Currently, her area of research deals with cells mechanism that determines or alters cell fate. She has received various outstanding fellowships and awards. Noted among them is DST-SERC Young Scientist Award in 1990.
Thank you for being with us in this show. Let me start with this very simple question, which I think many of our viewers would have in our mind. What is the stem cell? We know about skin cell, we know about blood is a cell, so blood cell. We know that brain cells are called neurons. Is it some kind of a special cell that lives in some place?
Right. So it's actually probably a little bit of everything that you said, but stem cell, I think, is most simply understood as a cell which can give rise to many other types. Right? That's one component. So you can have stem cells which give rise to your brain. You can have stem cells which can give rise to skin, to your blood components, to different body organs. But an important element about stem cells, and which has also been the challenge to understand them, is that they continue to be in you and me and in children, but they are very specialized and they maintain what we call their identity. So they stay as stem cells throughout much of our lives.
Okay.
And that also brings us to what is one of the challenges today of understanding how does a cell live in an environment which is like the liver or the brain or skin and maintain itself as a stem cell.
So let me try to kind of understand what you have said. Say, for example, if I have a bruise here, a new skin comes.
Yes.
So they are all skin cells.
Yes.
And they are all generated from the cells around this area.
Which is the stem cells.
Which is the stem cells.
Which is sitting in the area of the skin.
In that area.
Yes.
And they produce the skin cell because it is in skin.
Yes.
But the same kind of cell, for example, sitting in some other part, let's say liver, produces a liver cell.
Yes, but what we think is that you could take a stem cell which is sitting in the liver and make it make stem cells in the skin and therefore regenerate skin. And this is the hope of stem cell therapy.
Okay.
Right? When you take a piece of your skin or you take a little bit of your blood, like the doctor does in the clinic, and then you use that using technologies we have today to make what are called induced pluripotent stem cells. Right? So this is the iPSC that people hear about a lot. So this is you're actually making a stem cell in a dish. And what we hope today and has been demonstrated is that you can take this iPSC and actually make different kinds of tissue out of it.
So if you say that there is a stem cell sitting there, how does it know that I have to produce skin and not liver? Suppose if there is a bruise and then it produces a liver.
Very nice question. So that's exactly what we're trying to understand, right? That how do you stay a stem cell? And how do you know when you're in the skin, you should only make skin if I have a cut and not start making another kind of tissue, which is like liver, right? And so that's what institutes like ours and stem cell researchers the world over are actually trying to understand. That how do you stay quiet and when you get active, only become what you should be. So how do you respond to an instruction? And so therefore only be a skin and not be liver or not be brain in the wrong part of your body. So very, very simply, I think that's the core of
okay, very interesting point. But suppose as a kind of a lay person, if I'm going to ask you this question, that your institute or other institutes around the world what are the kinds of questions that they are asking when everybody says that stem cell research? Obviously, there are many institutes, many researchers. All of them would not be asking only one same question, right? So, what kind of different questions?
So, what are the kind of
questions which are being?
Yeah. So, what kind of questions you want to ask is how might you use stem cells for regeneration, right? Or let's take a more everyday example. So regeneration is, of course, if you have damage or amputation or you've lost some part, right? And you want to remake it. But that's the bottom line of all regeneration. But for instance, we know that there are many conditions, life conditions like diabetes or others, which give you what are called festering wounds, which don't heal. Now, for instance, we have a group at our institute which is trying to understand how do stem cells contribute to wound healing and repair. So the cut on your skin is sort of mimicking in a very small way, in a very controlled way, the very big damage you can have, for instance, if your limb is lost and you want to regenerate your limb.
So basically a cut is kind of a very small thing of, let's say, if there is a cut in the...
As opposed to a cut across the arm where you might...
So this heals, but that doesn't heal in a same sense. So you're trying to see how does...
So one is trying to understand what are the rules that operate at this level. Then you know, there is this other issue of like cardiomyopathies, for instance, and cardiac disease. Very often that is because there is a failure of the cardiac tissue to repair itself. Right? So you want to try and understand what is lying behind this. And you do this by trying to take the tissue, take the stem cells, and try and understand the process of building a heart. So there's a component of what we call developmental biology, as opposed to just regeneration, which is when you've lost it and you want to build it again. So these are, you know, the sort of the challenges at the level of research. Then there are issues of the clinic, where you want to try and generate these for therapy. You want to try and you learn how to store them, how to make what you want out of them. These are sort of
very
burning
interesting.
questions.
Let's take two examples. I mean, if we look at life form, for example, plants. You pluck a leaf, the plant is not, in that sense, greatly damaged. Most likely it will regenerate its leaf. Even among, let's say, animal life form, for example, lizard, if it loses its tail, it can regenerate. Are we saying something like that's a kind of direction that perhaps the regeneration medicine could take us to?
It's interesting. I'll just say that the loss of the tail of the lizard will regenerate only once. So there is a rule there which you want to understand.
So even in a thing, it will be doing only once.
It does it only once. It can't do it again
Why
and
it
again.
can't do again is also an important question.
So it's a question that you want to, so this is, you have to want to know how it works. But I think that's fundamental to all biology, right? You want to know how does a thing work before you start thinking of how you might be able to use it. And for, I mean, obviously with stem cells, the proximity to medical benefits and health benefits is so close that it would be unnatural to work in stem cells and not think about how to use it for medicine. How might you apply it?
One, again, a simple question, like for example, if you take, let's say, a stem cell from skin, and you say that you can't distinguish from that cell to a stem cell that is perhaps taken from liver.
We can today.
We can today. I mean,
yes,
there is
we know that in our bodies, yes, they are in different niches. There will be what we call identifying marks. It's like a postal code, which says that you live in this part of a city as opposed to another part of a city. There is a postal code.
So the stem cell has a kind of a stamp on it,
on
which
the
says
skin,
that this stem cell is actually coming from skin.
To some extent.
To some extent. So which means that the stem cell from skin cannot be used to grow a liver or vice versa.
That's what I'm trying to tell you, is that today that is possible, because when you make the induced pluripotent cell, you take a piece of your skin or blood and you actually make it into a stem cell, which you can drive to be either go to skin or go to liver or heart or muscle.
So major challenge would be how to make it to grow into a full liver, so that perhaps if you want to replace, you can replace.
That's
precisely. That's a very interesting point. With this note, we'll take a very short break. Don't go away. We'll continue this very interesting discussion. Keep watching Eureka. Welcome back to Eureka, and then we are having a very interesting discussion with Dr. Apurva Sarin, who is the Dean of Stem Cell Research Institute and Regenerative Medicine in Bangalore, which is part of Department of Biotechnology. This institute, when was it formed?
2009. As a partnership between the, the Department of Biotechnology and the Department of Atomic Energy, TFR, essentially thought that it was time that India took a lead in this direction and set up this institute.
So it's one of the kind in India?
It's one of the kind,
Okay. How big is the institute and what kind of major challenges that you are addressing in this institute?
So we are currently about 14 faculty level appointments, and we have about 2 to 3 senior appointments. So it's an institute which perhaps may be a little bit bigger than what might have thought for something that only started in 2009. But as I hope we'll go on to discuss, part of the reason we could do this is because we built the institute in a campus which already had another mature space.
Okay.
But to tell you a little bit about the kind of challenges, I think our challenges are not sort of not unique to us. I think they are faced by every small institute. We've again been sort of lucked out a bit because of where we are located and also of course the larger culture of Bangalore. But the usual issues are of setting up to identify questions that one would see would be meaningful in the long term. Because I mean, this is more of a personal thought. I think one of the issues that one has to be prepared as a biologist is to be able to ask questions which you might not resolve in your own lifetime, right? You take on a question that is prevailing and then you want to try and address that, but you might not know what the full outcome would be.
Yeah, but you start the journey.
But you start the journey. And at InStem, what has been really exciting and different is that we have built what are called themes of questions. So we have groups of researchers who come together, work as teams. These are all independent faculty, but who've decided to come together and put their mind.
This is what you have done in InStem.
In InStem.
So can you tell us what are these groups? Which are the kind of questions?
So there's a group which is looking at wound healing, which is broadly under the umbrella of inflammation. We have a group which is looking at cardiac disease and fibrosis and looking at cardiomyopathies. This is a prevailing problem in children and it's a question of renewal and repair. We have a group which is looking at brain function, and that's a very interesting group on, we call it the Group for Brain Development and Repair. And that has also led to a larger partnership with our sister institute NCBS on this campus and the NIMHANS, which is the National Institute of Mental Health and Neurosciences, and maybe we can come back to that. Let me tell you about the other groups on campus. Then we have a group which is just developing, not just, but whose focus is to develop technologies. And these are technologies which will allow us to ask questions at a more advanced technical level in all of the other groups.
Can you give 1 or 2 examples of what kind
so
of
there are
technologies
people
you are
there
looking
who
at?
are studying, for instance, how do you probe a single molecule inside a cell, which may be a stem cell, which will be of particular interest to us. So we have a group of people who are doing that. We have a group which studies what are the sugar signatures on your cell surface, because we know today that the sugars on the cells are actually different and can determine the way they behave. So keeping in mind that your interest is to understand how is regeneration, repair brought out, what is the role of the stem cell, what is the role of surrounding tissue, you want to be able to understand everything that constitutes the environment, right? And then finally, the group that I'm in is actually one of the younger groups in the sense that it started more recently, and it's a group which studies basically how cells determine what they are going to be.
Okay, so how is it going to determine?
Yeah,
it's going to be
why are
skin
you
or
a stem
liver
cell?
or,
Why are you this? Why are you that? What is sort of your metabolic and biochemical signature? These are the things which define cells in chemical terms.
In the kind of groups that you described, there is one which kind of interest is that this collaboration that you have with NIMHANS. Can you kind of elaborate what this is?
So it goes back to the fact that we are set up in a campus which has NCBS, the National Center for Biological Sciences. I was a scientist there myself, grew in fact my early career there entirely, which is today 25 years old. Now over the course of these 25 years, NCBS has built connections with other institutes in Bangalore and otherwise, but with the clinicians at the National Institute of Mental Health, particularly clinics which are looking at what we call multi-gene disorders. These are like disorders like schizophrenia, like addiction, bipolar disorders. These are complex disorders.
These are not caused by single gene problems.
Exactly.
For example, sickle cell anemia is largely a single gene.
Like Fragile X, we think we know a protein that is responsible, a gene that is responsible for it. Not so for these much more complex disorders, which are also widely prevalent, right? And so, because of the relationship which has been built up with NIMHANS over these years, we have today, with the support of the DBT, we have set up a joint program of scientists at the Stem Cell Institute, at NCBS and NIMHANS to actually garner material from patients and look at not only just their genetic signatures, which are very important today to understand something, but also generate stem cells and see if we can try and understand, sort of unravel and open up and see what might be the factors that go into these kinds of conditions. So this is a particularly exciting venture, because this is truly something which is going to have to last for at least the next 10 to 20 years to come together. It's been possible because we have a group which is committed to wanting to understand disorders of the brain. And we had a group which was led by my colleague Dr. Shona Chatterjee, with collaborations in Edinburgh, to understand Fragile X and intellectual disabilities. And so, what this actually says is that if you nurture efforts in a campus over a period of time, make a commitment, you can actually come up with something which would not have been feasible unless you had done a lot of groundwork.
We are having a very interesting conversation, but then we need to take a very short break. After the break, we'll continue to discuss with her how this institute came about, promoted by NCBS, how this new idea came into existence. We'll discuss these issues after a break. Don't go away, keep watching Eureka. Welcome back to this episode of Eureka, and then we are having a very, very interesting conversation with Dr. Apurva Sarin, who is the Dean of Institute for Stem Cell Research and Regenerative Medicine in Bangalore, which is an institute under Department of Biotechnology. Before the break, I was just pointing out that this institute came about, though if it is under Department of Biotechnology, but essentially as an offshoot, or perhaps we can say that the budding done by NCBS, National Center for Biological Sciences. Why anybody thought that such an institute should come, and that too just across a stone's throw away from NCBS?
Yes, so it's actually not even a stone's throw. We are connected. We are just a connected building. But I think this is a very interesting question, because it builds out this sort of style of approaching how to address scientific problems in the longer term. The NCBS has developed investigator-driven programs of research. And what was felt for the Stem Cell Research Institute, and I think it was not just because it was stem cells, but that NCBS had the space and the curiosity to nurture an effort which might be more directed towards an application-oriented kind of research.
What people might call as translational research.
Kind of. Yeah, I'll say application and not translation, because we are not directly associated with a hospital on the campus. NCBS has obviously already had an effort where they have started looking at working outside of their boundaries, and they are asking questions in ecology and the ecosystem. And you should think of this as the clinical space, which was sort of untapped and untouched. And also the fact that the campus had a wide bandwidth. What I mean by that is that there is work on single molecules here, and there is also work, molecules that you can't see. And then there is work at the scale of an entire ecosystem like the savanna.
That's the kind of work the NCBS was doing.
Yeah, this is what NCBS is doing. And so what bringing InStem as a program actually into this campus has allowed InStem to tap into this very rich intellectual resource. Not only, of course, the facilities and the capabilities, because as InStem built, it already had things in place which it could benefit from. And I think it is this culture of collaboration which is very, very important and unique today. So at a very small way, we'd call a microcosm, InStem has teams of research, but as a larger unit, it is integrating and interacting on this very, I think, it was a very important sort of arrangement for InStem, but I think equally important for NCBS, because Shona Chatterjee, for instance, is a researcher at NCBS. but it allowed him to leverage what is available at InStem to drive this program. I too was a researcher here, but now I am associated with InStem. So, there are possibilities you can explore through such arrangements.
Yeah, very interesting. Let me kind of shift the gear and then touch upon some other areas. Like, for example, one thing that I'm curious is, how did you land up in this particular area? What made you passionate about stem cells?
Right. Yeah, good question, because anybody who knows my work knows that I don't really work on stem cells. Okay, so good you asked. But I've had an interest in trying to understand how cells are made, how do they live, what controls their life cycle. And my group has been asking this question in a cell type of the immune system, which is the T cells, right? And so the question that I am myself interested in is asking, how do you regulate number? So how do you know that, how does your body know that you stop making so many cells today? And that has been much of what my training has been throughout my PhD, postdoc, everything. And what InStem allows is a space where you can now ask, how general are the principles that you are exploring, right? Can you address this more broadly to other cell types away from the T cell? So I think to me that was what was exciting, and the possibility of working with other colleagues who are interested in different aspects of this same problem. So we have people who are trying to understand how does the blood niche remain as blood and how is the niche built, and that is a young colleague of mine, Tina Mukherjee. Also at InStem, there are people who are trying to understand how cells make food and how do they utilize their resources, how do they deal with less resources. These are questions which you can apply to stem cells as much as to other systems. And so,
continuing this same question, in some sense, let's say in terms of time, much even earlier in your life period, you would have been in school,
yes,
+2, yes, then come through for graduation. How did you, or what made you think that you should go into research line? I mean, people would be looking at various other options.
Yeah.
It's not that everybody comes across that the potential or possibility of being a researcher. How did this happen?
Well, I think a lot of it is to do, as in many cases, but not always, is to do with the home environment. I think if you're brought up in a family of professionals, you tend to think of that as a career choice. And it's not as wide as it was today, but it can be pretty wide. But I think it is also in wanting to know how things work, and the fact that biology appealed to me. And I think as you train, you find an area of biology that interests you more, and then you nurture that interest further.
Is there any incident or any anecdote in your life which actually kind of motivated you to be curious? Something that in your childhood or in student days or in college?
I'd say very good biology and chemistry teachers in high school.
Okay.
I have to think about it. I can't think of an incident, but I think it is more of an output of an environment that you are in, that you are sort of placed in, for which I am very fortunate, I'd say.
Yeah. Another question that many of our audience, perhaps because in this country, it's not easy for a woman,
yes,
to build their own careers. I mean, what would you say? I mean, like, what kind of challenges you faced? And many of our audience are also young women. What would you say to them?
Yeah, I would say that you must trust your instinct and do what excites you the most. And whether it be biology or research, I think that's the choice to make. But having said that, I think it is not so easy for all kids, whether they are boys or girls, to sometimes persuade their parents that research is a career choice. And, I wish there was a simple answer.
Yeah, of course, yeah.
And of course there isn't. But I'd say stay true to what you want to do. Find good teachers, find individuals who can persuade those who are not persuaded about your inclinations, and work with that. I was particularly fortunate, as I said, and I didn't have to actually struggle to enter education and this profession. But
because your family was kind of...
Yeah, everybody is not so fortunate. Yeah, and yeah. I mean, it's a question of choices, and I think there is a lot of possibility today. And I understand where parents are concerned about, because there is an uncertainty in our in this choice. Yeah, because you're entirely curiosity, entirely driven by going into the unknown. But I think that the structures today are very supportive.
Like there is no answer in the last page.
There's no answer in the last page, but I think there's wonderful promise, and it's very exciting.
Very interesting. Let me start with the point that I made in the beginning of the program, that a lot of people look to stem cell research with hope, but there are also few critics who kind of, let's say, look at it with certain negativity or perhaps give caution. What are the issues? What are the challenges?
I think this concerns almost any kind of biology which is approach which is interventionist. The fear, I think, is that what if you make something that you cannot control? Yes, and I think that pervades a lot of our thinking in society today. I think as scientists, we have to take perhaps much more responsibility in our ecosystem in India to explain perhaps what is the possibilities and what are the cautionary notes. I think trying to pretend that there is nothing to be fearful about is not wise, but there is so much promise, and if done with discipline, I think there is a lot to be accomplished and that one can get. So the fear again of stem cells is pretty much the fear of anything that you are making in the dish, as to how do you know what this is going to be. So one has to deal with ignorance, but also the real fears, and explain what are the kind of disciplines that operate, so that you will not be actually making monsters in a dish. Yeah.
You are talking about this monsters in a dish. That's a very important point. But then there are also a lot of people who talk about regulating stem cell research. What's your take on it?
Regulation is necessary. Yeah, but I think blocking discovery for the fear of finding something that would make us uncomfortable is something that I would not advocate. And I think that's why I say that regulation, but informed regulation, is needed. So there are certain constraints on research with embryos today, which we've all read about. There's been in the news, and much of it is understandable. I think there is scope there for greater discussion and greater understanding of why people think that you should be permitted to research beyond the period specified. Having said that, I should also say, since this is about an effort in our own country, that we have very informed colleagues who work in this process, who are trying to set up regulations that govern our research. But there are some decisions, where we have to engage with the international community, and I think that is an important ongoing process, and it will continue. Yeah, it's going to be a bit of a struggle, but it's.
I think this is a very interesting and important point, that informed regulation, not just blind regulation. Restriction. Informed regulation and not blind restriction, and then that's a note in which we need to end today's discussion for want of time. We could have discussed quite a lot on this interesting area of stem cell research, but then we will end this conversation here. Keep watching Eureka. Next week we will come up with another episode with a conversation with another scientist. Keep watching.